Hi, welcome back to part 3 of chapter 44, Osmoregulation and Excretion. So we just got done talking through the whole process of urinary system and how it does go through the excretory process. Hence, now we're going to end the chapter with adaptation of vertebrate kidney. And like I said, I shouldn't take longer than 20 minutes or 15 minutes or so, but I don't want to drag the part 2 into hour and a half. So that's why I want to make sure even though we don't have much left, I want to make it a part 3. All right, so the ability of mammalian kidney to conserve water is the key adaptation for living on land in that sense, because we just talked about how we can, modulate it to absorb more water or less water, more solutes or less solutes. So for human, osmolarity of blood is about 300 units, but kidney can actually excrete urine up to 4 times as concentrated as our blood, which is really cool, which means we can excrete 1,200 units of units of the osmolarity of the urine itself, which is pretty cool. And that's mainly due to the presence of juxtamedullary nephrons, which is what I talked about, how they have a long loop of Henle, hence they can allow for a lot of water reabsorption, hence a lot of salt um, reabsorption, because salt and water always go hand in hand, because water always follows salt, right? I talked about that. So wherever you have salt reabsorption, you always have water reabsorption, because water will just follow wherever there's more salt. All right, so the differences among species in the length of loop of Henle and relative numbers of juxtamedullary and cortical nephron is what fine-tunes their osmoregulation for specific habitats. For example, a species of Australian hopping mice, they can actually produce urine 25 times as concentrated as the animal's blood. Now, in their case, they actually live in the deserts. So it makes sense that they can do even more concentrated urine, because they cannot afford to lose any more water than they already is losing when they don't have access to a large body of water in the deserts. So I guess that's why the environment, like we talked about all along, selects for the traits that is best suited for that living and thriving in that particular environment. So mammals that excrete the most hyperosmotic urine, I already said, is Australian hopping mice. There's also North American kangaroo rats and also other desert animals. That's a common thing about these guys. They live in the desert, don't have access to a lot of water, so they have a lot of juxtamedullary nephrons, with loops of Henle extending even deeper into the medulla than humans do. So that's why they can reabsorb a lot of salt and and water. And so they have a very steep osmotic gradients within the kidney, thousands and thousands of units at the bottom probably. And mammals that live in fresh water, such as beavers, muskrats, and your any other aquatic animals that you can think of, have mostly then cortical nephrons, because they live in fresh water. They don't need to have, produce, concentrated urine. If anything, they need to release diluted urine, because they're constantly exposed to water. So they have very low ability to concentrate urine, and that's because they rarely face problems of dehydration when they're around water all the time. So this particular example of mammal, which is really interesting, it's a South American vampire. They call it vampire bat, because it drinks the blood of its prey and doesn't suck it. It actually laps it up with its tongue. But the versatility of this particular mammalian kidney kind of illustrates how, diverse, environments that our kidney can do for different species and different habitats, or even the diet they have. For this particular vampire bat, they usually, like I said, feed on blood on large birds and mammals. And they usually consume as much blood as possible when it finds its prey, because it takes a while for them to catch a prey, especially when they're this tiny little bat trying to find the bird that they can subdue and also kill with their tongue, obviously kill with their teeth, and then, that animal will be there for them to lap up the blood. So once they get a prey, they would lap up as much as they can, and they can eat up to 24% of their body mass. Excuse me, not 24%. They can eat up to more than half of its body mass. I was reading a line lower, more than half of his body mass, which is, I was like 24. No, it's a lot more than half. So it's a lot heavier after it has eaten its prey. So when it's heavy, but remember, these are still bats. They need to fly back to their cave. So somehow they gotta lose all that weight very quickly. So the kidney has evolved and adapted to this particular diet where as the bat is feeding on all this blood, the kidney will excrete large volumes of dilute urine for as it's feeding on it. So that's pretty interesting, which means wherever that bat feeds, you will see a whole pile of fluid in that area of its urine. They say they can urinate 24% of its body mass per hour, which is really, really fast peeing. So as they're done feeding, then they should also already have lost all the water weight that they have just ingested with all that blood, and they can then fly quickly back to its cave. Or else, with 50% more body mass, they cannot ever fly back to their cave. But because of this ability for their kidney to quickly switch up and urinate as much water as you can and retain all those protein it's got from the food, then it can fly and go back to its cave. Now the problem is once it goes back to its um, cave, then all the protein from the blood starts to be digested. And remember, we said when you metabolize macromolecules, you will form a lot of urea. And in this case, they form large amounts of it because that's a lot of protein they have gotten from the blood. And so in the cave, it actually has no access to water, so they cannot even dilute that urea and pee as much of that their urea out because they already pee all that volume of water out when they were feeding on the prey. So now the kidney is able to shift to producing very small quantities of highly concentrated urine. We're talking about 4,600 units of osmolarity, and they then dispose of the urea through that tiny amount of liquid. So whatever water they can conserve, they can do it as much as possible back in the cave. So because of this really huge range of osmolarity of urine that these bats can pee, this is what makes them very interesting to talk about in the urinary system because they can adapt according to their particular diet. All right, so there's differences amongst other species as we continue through these last two slides. Most birds, including ostrich, actually live in dehydrating environments. So their kidneys usually have a lot of juxtamedullary nephrons like mammals. Remember that no other species other than mammals and this birds have juxtamedullary nephrons. Other guys don't really have it. So but nephrons of birds in the loop of Henle actually extend less into the medulla than mammals do. So that means they cannot absorb as much water as we or as our mammalian kidneys because their loop of Henle is not as long. Remember, it is the bird's main water conservation adaptation is having uric acid as its metabolite or nitrogenous waste. So that means they don't need to eliminate urea in liquid form. They, relieve their uric acid in that semi-pasty form, in solid form, so that they can conserve as much water as possible. So it's okay that their loop of Henle don't absorb as much water. There's this other adaptation that backs it up. And then you have reptiles, which only have cortical nephrons in their kidneys and will produce isoosmotic or even hypoosmotic urine compared to their own body fluids. So, given in such situation, it is the epithelium of their cloaca. Remember cloaca in the sharks? Same thing here. It's an opening where urine and feces are excreted. They can actually conserve fluid by reabsorbing that water from these wastes within the cloaca. So probably they have some sort of kind of like the rectal gland we talked about in the shark, but instead of reabsorbing salt, excreting salt, it is for reabsorbing water in the reptile's case, just because they're losing a lot of water through peeing. And then you have freshwater fishes, which are hyperosmotic to their environment. We already talked about this. They will produce large amounts of very diluted urine. Their kidney is packed with cortical nephrons because it relies on absorption of ions from the filtrate within the distal tubules. And then you have amphibians. Amphibians has the capacity to adapt to land and water. So in freshwater, they tend to excrete dilute urine, while skin accumulates salt from the water by active transport. So within water, that's what they do. Just like a freshwater fish, they would, excrete a lot of dilute urine, and then they can retain the salt through their skin. On land, then they kind of act like mammalian, where they conserve body fluid by reabsorbing water across the epithelium of the urinary bladder. Then, of course, they will also pee less, just like human beings do. But of course, we cannot absorb water across our urinary bladder, but they can for frogs, amphibians. Last but not least, you have marine fishes, which we again already have talked about that. From the ocean, they have fewer and smaller nephrons. Some even don't even have the distal tubule. And for some marine fishes, they even lack the glomerulus too, because they don't need to filter stuff very much. There's already a lot of salt in the surrounding. Water is getting removed from their body already. You don't need very high filtration rate. So there's a very low filtration rate because there's no urine that needs to be excreted. You better not excrete a lot of urine because you need all that water for all the salt that's coming in. Excuse me, oh, not that. Well, there's all that salt coming in, and also you're losing water. So that's why you don't pee as much. So main function of these marine fishes in ocean is one thing is that they need to get rid of their divalent ions. I kind of talked about that earlier in the chapter, that they need to get rid of magnesium, which is a divalent ions, means that they are a 2 positive ion. So calcium 2 plus, magnesium 2 plus, sulfate 2 plus. So they need to get rid of those. So somehow they cannot survive with a high concentration of these divalent ions for some species, and they would get rid of them through proximal tubule by secreting it into there. So bony fishes will rely on chloride cells as well, which is what that crash course guy was talking about. Chloride cells. Here it is, and they're in the gills, enable these bony fishes to secrete sodium chloride salt. That's why they call chloride cells. And last but not least, in this chapter, we will, kind of go over the gist of the hormonal control within mammals. How do we regulate our kidney functions? So one of those we already talked about, which is ADH, antidiuretic hormone, released by your posterior pituitary gland to increase reabsorption by water and by inserting the aquaporin in the collecting ducts. This, we already talked about, results in smaller volume of concentrated urine because you reabsorb a lot of that water back. Now, if there is any mutation to this particular protein, either you cannot produce ADH or you inactivate ADH receptor genes such that you have nothing for the ADH to bind to and cause a effect, you can actually have severe dehydration and solute imbalance due to production of large amount of dilute urine constantly all the time. There's no regulation of reabsorption of water whatsoever. So you just keep peeing whatever water you have, and there's no conserving. So this condition is called diabetes insipidus. That has nothing to do with blood sugar. It just goes according to what those words means. Diabetes means I think it was to pee, and then insipidus mean no taste. So this is urine with no taste, something like that. And then so that means it's very dilute urine. Probably diabetes mellitus. Mellitus means urine with some sugary taste, and that is what that signature of diabetes mellitus patients is. Their urine is full of glucose. But there's nothing, in diabetes insipidus, nothing to do with blood glucose, just not seeing ADH, and you're not conserving water at all. And then so when you have situation where it's not talking about osmolarity lost by that ADH can rescue, but what about when you are losing blood volume without affecting osmolarity? So if you all of a sudden got attacked and got a big wound, got into a car accident, now you're bleeding like crazy, or you have severe diarrhea, you're just losing a lot of the water in your body that way. So the kidney actually or endocrine system actually have a whole circuit called the renin-angiotensin aldosterone system, or the RAS, RAAS, the RAS circuits. Though this respond in any very low drop in blood volume and blood pressure, like I said, such as major wound or the severe diarrhea, and they will increase sodium reabsorption, which in turn will increase water reabsorption. How it does it is that once your body senses that there is lower blood pressure and lower blood volume, kidney will release an enzyme called renin, which will activate angiotensin, then stimulate your adrenal glands to release aldosterone. Aldosterone is the hormone that increases reabsorption of sodium ions through the distal tubules and collecting duct. And of course, when there's a reabsorption of salt, I always say salt and water goes hand in hand. You reabsorb water as well by osmosis because water follows salt. Both case scenario, you have increased your blood volume back up and build your blood pressure back up to homeostasis. So even though you're dying with, bleeding a lot, your body will do its best to try to get your blood pressure and blood volume back up as much as it can. Of course, the longer you're bleeding, the less likely your body won't be able to keep you in homeostasis anymore until at least maybe if you're just lasting for that couple more minutes before ambulance arrive, your body can keep you alive for that, couple more minutes by the RAS circuits. And then you have AMP, which is an atrial natriuretic peptide, hormone that is released by the heart for when blood volume and pressure is too high. So AMP works in antagonistic or opposite relationship with RAS and ADH. So it actually inhibits the release of renin, which then opposes the action of RAS. RAS is for increasing water reabsorption and salt reabsorption. AMP is stop all of that. Don't reabsorb water. Don't reabsorb salt. Let all those water then drain out of the body. That's what AMP is. And that's what you have to do when you have too high of a blood volume is let some of that water go. And that's all I have for you for chapter 44 on osmoregulation and excretion. Lastly, we have the practice question and then we'll be done. Osmoregulation is what? It is process by which animals control solute concentrations and balance water gain and loss. Which of the following is not an adaptation of the vertebrate kidney to diverse environments? Marine bony fish have more and larger nephrons. We did not talk about that at all than freshwater fishes, so that's why it's wrong. But good to look at the other four choices as well, because that means the other four choices are something we talked about and is true. Which animal are being studied as examples of an adaptation called anhydrobiosis? We talked about this, saw a video of this. Tardigrades or water bears. What effect would you expect the drug furosemide, which blocks transport of sodium ions and chloride ions in the ascending loop of Henle, to have on urine volume? Think about what ascending loop of Henle in general causes. It should be increased in urine volume because it blocks the transport of sodium chloride. So it doesn't cause, if you don't reabsorb sodium chloride, you don't reabsorb water. So all that water just going to go into the urine. So that's why increased urine volume. Animals that can reproduce exceptionally concentrated urine, like they can produce, should be expected to have nephrons with longer what? They have exceptionally concentrated urine. What should they have longer of? Should be your loop of Henle, right? 1 and 2. In the kidneys, which hormone is most effective at causing increased number of aquaporins to be present in the collecting duct, and what is the source of this hormone? So we talked about this at the end and also before in the middle. ADH, posterior pituitary gland. Which of the following statement about AMP is incorrect? Something false about AMP. A, because the wall of heart release AMP in response not to low blood pressure and low blood volume. It is released in response to increase in blood volume and pressure. ADH or RAS pathway is for decrease in blood pressure. Under which circumstance would reabsorption of water from the kidney filtrate back into the bloodstream be most effective? So if ADH and aldosterone were present in the circulation, they are both, ADH and aldosterone also work hand in hand, just like water and salt work hand in hand. How does the RAS pathway respond to in the case of excessive ion of both salt and body fluids? Excessive loss. It responds to the drop in blood volume pressure by increasing water and sodium ion reabsorption. That's the whole point of RAS, is to cause sodium reabsorption and hence water. And that's all I have for you.